trisomic rescue via allele-specific multiple chromosome cleavage using CRISPR-Cas9 in trisomy 21 cells.
Wow, that's a mouthful.
I know, it's like, okay, what does that even mean, right? But basically what we're talking about here is Down syndrome.
Exactly.
A condition that, as many of you probably know, arises from having an extra copy of chromosome 21.
Right. Trisomy 21.
Trisomy 21.
It affects a significant number of births, and while there's been so much research focusing on understanding its effects, this paper tackles a very, very fundamental question. Could we actually eliminate that extra chromosome from the affected cells?
Wow.
And historically, that's been a very, very less explored avenue of research.
Right. Like, can we just correct the underlying cause right at the source?
The root of it.
Which is why this feels so significant, right? It's exploring this completely novel approach using some very, very advanced technology.
It is.
So for you, the listener, like, our mission here is to distill those crucial findings of this research. Like, what did they actually do? What did they find out? And then, like, most importantly, what could this mean for the future of ways we address Down syndrome?
Yeah, and we're committed to keeping this accessible no matter your background.
Exactly.
Whether you're, deeply scientific or you're just interested.
Right.
We want to make sure you can follow along.
Absolutely. And it's important to keep in mind that our source here is a very detailed scientific report.
Yes.
We're going deep into the specifics of, like, their experiments, what they observed, all that good stuff.
Exactly. The nuts and bolts of it.
The central challenge that they're really trying to address here is this lack of effective methods to actually remove that extra chromosome 21 in people with Down syndrome.
Right.
So let's get into it.
Let's do it.
Okay. One term that you hear a lot in this kind of research is CRISPR-Cas9.
Yeah.
So in, like, the most simple terms, what is that?
Imagine a pair of really, really precise, almost microscopic scissors for DNA.
Okay.
So CRISPR-Cas9 is a genome editing tool that basically allows scientists to make very, very targeted changes to DNA sequences.
Oh.
So they can cut, add, or remove little pieces of genetic code at very specific locations.
It's like a find and replace almost.
That's a great analogy.
Right. But in this case, they're not just, like, changing a word in the sentence. They're trying to remove, like, an entire page. Right? Like, how do you even begin to do that with these molecular scissors?
So what's really innovative here is they're using CRISPR-Cas9 to make multiple cuts across that extra chromosome.
Okay.
And the hope is that all these breaks are going to make it so unstable that the cell's going to basically just be like, okay, I'm done with this.
Okay.
And just kind of discard it.
Wow.
And one thing to keep in mind is that previous attempts at this have been what's called allele nonspecific.
Okay.
Meaning they could potentially target any of the 3 copies of chromosome 21.
I see.
Without preference, which could be problematic.
Okay. Which leads us to, like, a really important part of this research. They developed this thing called an allele-specific, or as CRISPR-Cas9 strategy.
That's right.
So why is it so vital to be able to target just one specific copy?
Yeah, that's a great question.
Out of 3, right?
So this gets into some really fascinating aspects of chromosome 21 biology.
Okay.
Because while most genes on chromosome 21, kind of behave normally across all the copies, there are some genes that show what's called parent of origin dependent silencing or imprinting.
Okay.
And the paper gives a great example of this. It mentions the DS-CAM gene.
Okay.
And that gene is only active if it's inherited from the father.
Wow.
Additionally, genes like superoxide dismutase type 1, mitochondrial ribosomal protein L39, and SON.
Yeah.
These are all genes that can be expressed at different levels depending on whether they came from the mother or the father.
Okay.
So you can imagine that, if you just randomly eliminate one of the 3, it could have unintended consequences.
Yeah, I don't want to knock out the good copy.
Exactly. You need to be very precise in your targeting.
That makes total sense.
Yeah.
So how did they actually achieve this, like, incredibly specific targeting of just one chromosome?
So they developed this very clever phasing method to identify and target just a single version or allele of the chromosome.
Okay.
And for this particular study, they chose to target the maternal M2 allele.
Okay.
Out of the 3 copies.
So just to clarify, they're not saying that the maternal copy is always the one you want to get rid of.
Right.
They just randomly chose that one.
Exactly. It was just for this study.
Okay, got it.
So they designed their CRISPR system to recognize unique DNA sequences that are only present on that particular M2 copy.
Got it.
And that's really the key insight here. They can pinpoint and target one specific extra chromosome out of the 3.
Okay, so that's huge.
It is.
So they've got this incredibly precise tool.
Yeah.
They've got a strategy to target the right chromosome.
Yep.
So the next logical step is, okay, let's test it out.
Right.
What kind of cells did they actually use in the lab for this?
So they used induced pluripotent stem cells, or iPS cells for short.
Okay.
And these are cells that are taken from an adult and then reprogrammed back to an embryonic-like state, meaning they can become any cell type in the body.
I see.
They also use fibroblasts, which are connective tissue cells that were derived from an individual with trisomy 21.
Okay, cool. So the next question is, how do they go about finding those unique cut locations on that specific M2 chromosome, right?
Right.
It's like finding a needle in a haystack.
It really is. It's a very detailed process. They started by sequencing the entire genome.
Of the trisomy 21 iPS cells.
Right.
And then comparing that to a panel of disomic cell lines.
Yeah, so basically cells with normal 2 copies.
Right.
Where each cell line was missing one of the original 3 copies from the trisomy cell line. So that way they could really pinpoint which sequences were unique to the M2 allele.
I see.
And they found a vast number, over 15,000.
Wow, 15,000.
Unique target sites.
That's a lot.
It is. And a surprising number of them were concentrated in one particular region. At the end of the long arm of chromosome 21, called the Q subtelomeric region. But having this abundance of potential targets was really key to their strategy.
So how did they narrow down 15,000? Because that's a lot.
It is. They couldn't test all of them, so they focused on 56 targets within that Q subtelomeric region. And used a screening process with something called EGXXP plasmids. And these are essentially like test kits to see how well Cas9 could cut the DNA at those very specific sites. And this let them categorize the different sequences based on their cutting ability. So some would cut only the intended M2 target. Some cut both the M2 target and other similar sequences, which we call off-targets. And then some just weren't very efficient cutters at all.
I see.
So they ended up with 18 sites that had really good M2 allele-specific cutting activity.
So that brings us to they've narrowed it down. They've got their targets. They're ready to actually try to eliminate the chromosome using those selected sites.
Exactly. So what they did was they created these genetic vectors. And you can think of these as delivery trucks. That carry the Cas9 enzyme and the guide RNAs. And the guide RNAs are like GPS coordinates, directing Cas9 to the specific M2 DNA sequences. So they designed these vectors to target between 1 and 13 of those M2 specific sites simultaneously.
Wow.
And when they introduced these vectors into the trisomy 21 iPS cells, they observed that the rate of cells that corrected to the normal 2 copies increased as they targeted more sites to 13 cuts.
I see.
Interestingly, single M2 specific cuts at any of the 13 individual sites didn't significantly change how often the chromosome was lost.
I see.
So it was really the cumulative effect of multiple cuts that seemed to drive that chromosome elimination.
So more cuts equals higher chance of losing the extra chromosome.
Yeah, more or less.
Now, one thing that I noticed in the paper was this term repair gene inhibition.
Right.
So what does that even mean, and why did they do that?
So when Cas9 makes those double-stranded breaks in the DNA, the cell has natural mechanisms to fix them.
Makes sense.
And the researchers hypothesized that if they could kind of hinder these repair processes temporarily, it might make the cell more likely to just get rid of the whole unstable chromosome.
I see.
Rather than try to patch up all those breaks.
So it's like if there's too much damage, it's like, I'm not even going to bother.
Exactly. It's too much work. So they use something called small interfering RNAs, or SIRNAs. And these are kind of temporary silencers for genes. And they use these to reduce the activity of 2 key DNA repair genes. PLQ, which is involved in a pathway called microhomology-mediated end joining, or MMEJ. And LIG4, which is essential for non-homologous end joining, or NHEJ.
So we can just think of these as like the cells patching crews?
Yes, exactly.
Right.
NHEJ is like a quick and dirty glue job. While MMEJ tries to be more precise, but sometimes makes little errors.
So did temporarily, kind of hindering these crews actually lead to more chromosome loss?
Yeah, it actually did. They found that knocking down POLQ and LIG4 significantly increased the rate of chromosome loss across all the different numbers of M2 specific cuts.
I see. It's an average increase of almost 1.8-fold.
So like the key takeaway there is that just making cuts isn't enough.
Okay.
You also have to hinder the cell's ability to repair them.
Exactly. That's that second crucial step to actually losing the chromosome.
Okay, that's super interesting.
Yeah.
Now, they also compared their very specific allele-specific method to one that wasn't specific to a single allele.
That's right.
What did they find there?
So for the allele nonspecific targeting, they used CRISPR systems that targeted these repetitive sequences that are present on all 3 copies of chromosome 21.
Okay.
So they designed systems with a lot of potential cut sites,
Okay.
24, 49, and even 73.
Wow.
But surprisingly, even with way more cutting locations, these allele nonspecific systems resulted in lower rates of karyotype correction compared to the allele-specific approach with just 12 or 13 cuts.
I see.
And even more concerning is that allele nonspecific targeting was much more harmful to the cells, leading to a significantly lower survival rate.
So it's like quality over quantity.
Right.
It's not just about how many cuts you can make.
Right.
It's about where you make them.
Right.
And minimizing those off-target effects.
Makes sense.
Yeah.
So when a cell actually did lose a chromosome, how could they be sure it was the whole chromosome, and specifically the M2 allele?
Great question.
Right.
They use some really cool techniques.
Okay.
So first, they use something called FHH probes.
Okay.
Which are fluorescent DNA sequences that basically light up specific regions of a chromosome.
Okay.
So by using probes that bound to different locations along the length of chromosome 21, including the very end, they found no evidence of truncated chromosomes.
Okay.
Meaning it really seemed like the entire chromosome was being lost.
Right. Not just like a little piece breaking off.
Not just a fragment, yeah.
Okay. And then how did they confirm that it was specifically the M2 allele they were targeting? Because that seems tricky.
It is. So this is where it gets really interesting.
Okay.
They created something called 3-color reporter trisomy 21 cells. So they engineered these cells so that each of the 3 copies of chromosome 21, the paternal P, the maternal one M1, and the maternal 2 M2, would glow under a microscope.
Wow.
With a different fluorescent color.
Okay.
So that way they could actually directly see which chromosome was disappearing.
That's so cool.
Isn't that neat?
Yeah.
So when they applied the M2 allele specific CRISPR system with 13 cuts, the results were very clear. The M2 chromosome dominantly disappeared, as evidenced by that specific color just vanishing.
Wow.
And then they took it a step further, and they analyzed individual cell clones using short tandem repeat markers or STR markers, which are kind of like genetic fingerprints. And this confirmed the targeted loss of that M2 chromosome, resulting in a significant percentage of cells with the normal 2 copies of chromosome 21.
So that's pretty definitive proof that their allele-specific chromosome elimination is actually working.
Yeah, it's really remarkable.
Okay, so that's super exciting. They managed to remove the extra chromosome, restore the normal count.
Right.
But now the question is, what about any other unintended genetic changes?
Yeah, that's the big question, right?
You know, because we're messing with stuff here.
Exactly. So they started with a technique called G-banding karyotyping. Which is essentially taking a picture of the chromosomes under a microscope. And looking for any big structural abnormalities. And they didn't see anything obvious in the rescued cells at that level. But they wanted to go deeper. So they used whole genome sequencing, or WGS, to look for more subtle changes.
In the DNA sequence itself.
Exactly. So it's like zooming in to the individual letters of the genetic code.
That's cool.
It is.
So what did that more detailed analysis reveal?
So the WGS analysis found an average of around 2,100 genetic variants in the cells that had lost the M2 chromosome. And when they compared those to the predicted off-target sites for their guide RNAs, they did find a small number that matched.
I see.
Interestingly, most of these off-target activities seem to involve the guide RNAs binding to the non-targeted P and M1 alleles. But at the intended target locations on the M2 allele. Just with a single mismatch in the DNA sequence.
So almost the right spot.
Yeah, just a tiny little error.
But importantly, they didn't find any mutations caused by Cas9 in completely unrelated regions.
Exactly. That was really reassuring.
Right. It's not just going haywire.
Yeah, it seems to be sticking to the general area where it's supposed to be.
What about larger scale changes, like structural variations within the chromosomes?
Yeah, so they did find a few. Mostly within chromosome 21 itself. The trisomic cells that still had 3 copies after treatment had slightly more of these variations compared to the disomic cells.
Okay.
And when they looked closely at those 13 target sites on chromosome 21, they found evidence that in those trisomic cells, the DNA had actually been cut at most of these sites.
I see.
But then repaired.
Okay.
Which sometimes left behind small insertions or deletions.
So like a patch job.
Yeah, like a little bit of a messy patch job.
Okay. And then in the disomic cells, they found something really interesting.
Yeah.
Tell me about that.
They found evidence of cuts happening on the wrong allele at some of those guide RNA sites.
So even with really careful design, perfect specificity is still hard.
It's a challenge.
Yeah.
Oncology is messy.
Right.
It doesn't always follow the rules.
So how did they try to minimize those off-target effects?
Yeah, so based on all their analysis, they designed a new vector called refined M2AX66.
Okay.
And this one only used 6 guide RNAs.
Okay.
That had shown really good on-target activity and very minimal off-target activity in previous tests.
So like the best of the best.
Exactly. Okay. And when they tested this refined vector, it still led to a good rate of chromosome elimination, comparable to some of their earlier vectors that had more cuts.
I see.
So it seems like fewer highly specific guide RNAs might actually be more beneficial
Makes sense.
than just having a ton with potential off-target effects.
Right, so less is more.
Sometimes, yeah.
Okay, so they've made progress on reducing those off-target effects.
Yeah.
But what about the actual characteristics of these rescued cells?
Yeah, that's a great question.
Did correcting the chromosome number have any impact on their basic biology or gene expression?
So this is where it gets really, really interesting for understanding Down syndrome on a molecular level.
Okay.
They performed RNA sequencing.
Okay.
Which essentially gives you a snapshot of all the genes that are being expressed in a given cell.
Got it.
And they compared the original trisomy 21 iPS cells to their rescued development clones.
Okay.
And the analysis really clearly showed that the cells grouped based on their chromosome number.
Okay.
The rescued cells had a gene expression profile that was much closer to normal.
Got it.
Compared to the trisomic cells.
Okay. Were there any particular types of genes that showed big changes?
Yes. They identified over 1,500 genes that were upregulated or more active.
Okay.
And over 1,300 that were downregulated.
Okay.
Or less active.
Gotcha.
In the rescued cells.
Okay.
And when they looked at what these genes actually do,
this
is called a gene ontology analysis.
Okay.
They found that a lot of the downregulated genes were involved in metabolic processes.
Okay.
Like the breakdown of sugars, cholesterol, alcohol, things like that.
I see.
And some functions related to the mitochondria, which are the powerhouses of the cell.
Okay.
On the other hand, the upregulated genes were strongly associated with developmental processes, particularly development of the nervous system, the generation of neurons, and neurogenesis.
Wow.
Which is the birth of new brain cells.
That seems very significant, especially considering, the neurological kind of characteristics we see in Down syndrome.
Yeah, absolutely. So they even did a gene set enrichment analysis.
Okay.
Which looks at the bigger picture of groups of related genes.
Okay.
And this showed that the rescued cells had increased activity in gene sets that promote early development of the central nervous system. And the proliferation of neural precursor cells. Which are the cells that give rise to neurons.
Wow.
So that's particularly relevant because developmental issues in the brain are such a key aspect of Down syndrome.
Yeah, absolutely.
And while they did find a cluster of downregulated genes on chromosome 21 itself, the majority of these differentially expressed genes were actually spread across all the chromosomes.
Wow, so it's not just about chromosome 21.
No, it really reinforces the idea that having that extra copy affects the entire genetic landscape.
That's wild.
It is.
Now, I also noticed that they compared their findings to other research, too.
Yeah, that's an important part of any scientific study is to see how your findings fit in with the broader context.
Exactly.
So they compared their gene expression changes to a study that looked at iPS cells derived from identical twins where one twin had trisomy 21 and the other didn't.
Oh, wow.
And interestingly, they found a partial overlap of around 30%. In the genes that were differentially expressed.
I see.
So this suggests that while correcting the chromosome number obviously has a major impact, an individual's unique genetic background also plays a role.
Yeah, that makes sense. Everybody's different.
Exactly.
And then they also looked at where these genes are normally active in the body.
Yeah, so they found that the upregulated genes in the rescued cells were strongly linked to genes that are typically expressed in the developing nervous system.
I see.
Even in those early stage iPS cells.
Okay, that's interesting. So what about like the overall health and function of these rescued cells?
Yeah, so they assessed how quickly the cells were dividing.
Okay.
And they measured the levels of reactive oxygen species, or ROS, which are often elevated in trisomy 21 cells.
I see.
And can contribute to damage.
Okay.
So what they found was that the rescued clones showed a slight increase in their proliferation rate.
Okay.
Meaning they were dividing a little bit faster.
I see.
And a notable reduction in ROS production.
Okay.
Which suggests overall improved cellular fitness.
So it's like they're healthier.
Exactly.
Okay, so correcting the chromosome number is having all these positive effects.
Right.
Not just on the genetic level, but like, basic cellular health.
That's right.
So that's exciting.
It is.
So the next question is, they went beyond these early stage stem cells, right? They looked at more specialized cells too.
That's a really crucial point for, thinking about how we might actually apply this research one day.
Right.
So they wanted to see if this approach could work in more developed cell types.
Okay.
So they took their most effective vector, that M2ASX13, and tested it in skin-derived primary fibroblasts from the same individual with trisomy 21.
Oh.
And encouragingly, they observed a significant rate of chromosome elimination.
Wow.
In these more specialized cells as well.
Okay.
And then, even more remarkably,
okay,
they did an experiment in fibroblasts that weren't actively dividing.
Okay.
And still saw a pretty good chromosome elimination rate.
Wow.
So this suggests that the process doesn't necessarily need cell division to see it
work.
Which could have implications for future therapeutic strategies.
Exactly.
And importantly, they didn't find any cases of chromosome 21 being gained in any of their experiments, so it was always towards the normal number.
That's right. It was always a correction in the right direction.
That's cool. So this is all super promising.
It is.
So in the discussion section, what do the researchers say about the bigger picture?
So they acknowledge that identifying the specific genes that are responsible for all the different characteristics of Down syndrome is really complex.
Right.
Partly due to individual genetic variation.
Right. Everyone's different. And also this concept of aneuploidy-associated phenotypes.
Okay.
Which are basically problems that arise from just having an abnormal number of chromosomes.
Oh, I see.
Regardless of what specific genes are on that chromosome.
That's right.
They also highlight that this CRISPR-Cas9 approach is relatively straightforward compared to some previous methods.
Okay.
And it doesn't involve permanently inserting foreign genes into the genome.
Okay, that's good.
Yeah.
They also noted that they mostly saw whole chromosome loss rather than just little fragments being deleted.
Yeah.
Why do they think that might be?
That's a great question. So they suggested it could be related to this thing called trisomy bias chromosome loss.
Okay.
That can sometimes happen during iPS cell reprogramming.
Okay.
Basically when you're taking those adult cells and turning them back into that embryonic-like state.
Got it.
Another possibility is that the cells that lose the entire extra chromosome just have a survival advantage.
I see.
So they're more likely to grow in the lab.
Okay, so it's like a natural selection kind of thing.
Exactly.
Okay.
And they also emphasize that even with the allele-specific targeting, they did detect some small-scale alterations.
Right.
So we need to keep improving.
Right. It's not perfect.
Exactly.
Okay. And then they also talk about the importance of those DNA repair mechanisms.
Yes. So they reiterate that their finding about temporarily inhibiting those DNA repair pathways involving LIG4 and POLQ significantly improved the chromosome elimination.
Right. So knocking those out really helps.
Yeah. It really seems to tilt the balance towards getting rid of that whole unstable chromosome.
Okay.
And they also mention some other existing techniques.
Okay.
But they point out that their allele-specific method has some unique advantages.
Okay.
Particularly being able to target a single chromosome copy.
Right.
Like one of those 3 chromosome 21s.
Exactly.
And this avoids the risk of something called uniparental dysegmy.
Okay.
Which is where both copies of a chromosome come from the same parent, which can also be problematic.
Okay, so this is really the best way to do it.
Yeah, it seems like it so far.
Okay, and then they also revisit the gene expression stuff too, right?
Yeah, so they noted that their gene ontology analysis really provides evidence that correcting the chromosome number could potentially reverse that characteristic gene expression signature.
That we see in Down syndrome.
Right.
It's huge.
It is, and they connect the downregulation of those metabolic genes in the rescued cells to similar findings in a mouse model that has an extra human chromosome 21. They also link the upregulation of the nervous system-related genes to the neuroanatomical and cognitive differences that we often see. And then finally, they emphasize that their allele-specific approach is especially relevant to trisomy 21. Because the 3 copies of chromosome 21 in individuals with Down syndrome are often genetically distinct because of the way that extra chromosome arises during the formation of the egg or sperm.
Cool. So to kind of bring it all together, what's like the biggest takeaway from this deep dive?
So the major breakthrough here is that we now have a very clear demonstration that it's possible to efficiently use this allele-specific CRISPR-Cas9 to eliminate